So I told you about several ways to analyze gene expression, especially mainly measuring the RNA level, expression level of gene expression. And this is one example. You look at RNA molecules from a normal cell and RNA molecules from a cancer cell, and then you can see which genes are more expressed in cancer cells versus normal and vice versa. But all of these methods actually average. So all of these methods average about a large number of cells, thousands and sometimes millions of cells. And so at the single cell level, you may have actually great heterogeneity. And you have a yellow spot showing that on average, you have the same level of expression between the normal and cancer cell, but if you look at single cells, you may get something very different. So you do need that single cell resolution sometimes to really understand what's going on. So the method that I will introduce next is used to analyze gene expression at the single cell level. So FISH is the name, and FISH stands for fluorescence in situ hybridization. What is hybridization? If you have two DNA molecules complementary in sequence and they bind each other, forming base pairs, that's called hybridization. But DNA molecule can also hybridize against an RNA molecule, or RNA can hybridize against an RNA molecule. So that is a sequence mediated base pairing of two strands of DNA or RNA. So in situ means that actually you're doing this inside the cell without actually taking the RNA out of the cell. And of course, fluorescence is fluorescence. Okay, so this is a cell, and this is a nucleus. Now, human cells have a nucleus inside which DNA resides. But then once the RNA is made based on the DNA sequence inside the nucleus, RNA is usually exported out of the nucleus into the cytoplasm. And here the RNA will be translated into proteins by the ribosomes. So one version of the FISH method is shown here. So if this is your messenger RNA that you're interested in detecting, then you can design DNA probes, 20 bases in length, that is complementary to the sequence of RNA right here. Then you can also label it with the first molecule. So you have one dye per probe. But you can also design another probe that is complementary to a different section of the RNA, the same RNA, and you can do this like 50 times. Okay? So what it means is that if you kill the cell, you can make the membrane permeable to the DNA probes. So in principle, if you then add these 50 different probes to the cell, then one RNA molecule will bind up to 50 probes, and meaning that it can be decorated with up to 50 fluorescent molecules. And normally inside a human cell, if you have a single fluorescent dye is not bright enough to see because of a large background. But when you have 50 in one spot, you can see it clearly above the background. That's the idea. Then each mRNA molecule will be lit up, and this kind of signal amplification eliminates essentially false positives and false negatives. False positives why? Because usually if you have, let's say, just one probe, but labeled with 50 dye molecules, that's bright enough to see, but then they can also bind other things that are not specific. and that's called the off-target effect. When you have 3 million base pairs of DNA inside a cell, so 3 billion or 6 billion, then it's hard to avoid that off-target effect. But when you have multiple binding to the same RNA, that if one of them binds somewhere else, that's not going to be visible. So you don't get false positive, false negative, but each RNA molecule has multiple dyes, so it's clearly visible. So that's basically this method allows you to detect every single molecule of an messenger RNA of a given type inside a cell. Questions? So let me give you an example. So this is data from my lab. You can actually see there's a single cell here, and the purple is the staining of the DNA. So when you see a large block of purple, that means that you have a nucleus. So nucleus is stained with the purple dye. And then you see the green dots. So there are single mRNA molecules of one kind that we labeled using the fish probes. And what we did was that we actually used some small molecules to induce that mRNA production, meaning that at time equals 0, you add a small molecule so that as a function of time, the gene will be expressed. You'll see the number going up as a function of time. So this is one single cell, and here you can count all these spots, 45 spots in this cell. And then here, 1 hour later, it's not the same cell because you have to fix the cells to do this measurement. But you see similar number, but 2 hours later now number is much higher. And then there's 3, 5, 8, and 24 hours. So what you notice here is that you see the number increase, but the apparent brightness of each spot doesn't go up. That is also one evidence that we are detecting single RNA molecules. So in fact, you can quantify the intensities, at the beginning, early in time, and then you can measure the intensity of these guys and plot the histogram, and then compare to the intensity distribution later in time. They are identical to each other. Evidence that we are detecting single transcripts. Questions? And you can do this in 2 different colors. So here, again, the purple is the nucleus, the DNA staining. And then you have one gene labeled with green, shown here, and then another gene is labeled with red. The name actually doesn't matter here. You can see that red and green are clearly distinct. And you can count them. So you can count the expression levels of 2 different genes at the same time from a single cell. In addition, you may see that there are some bright spots inside the nucleus. This is what's called the transcription site. This is where the gene for that molecule is located, DNA. And then RNA polymerase molecules are sitting there, and they're making many, many copies of the mRNA. So this is a nascent transcription site. There are probably like 20 or 30 molecules there, RNA molecules there. That's why it's displayed. And eventually they will be able to export it out. The nucleus, the cytoplasm. Questions? So here's another example. You can actually do image the transcription sites for 2 different genes. There are actually 4 of them in this cell, but you'll notice that red and green sites are actually very next to each other. So that was quite interesting. And then until we looked at the genome sequence and where the genes go, and they're on the same chromosome, about a million base pairs apart. So maybe it's not surprising that they are close to each other. All right, so then this is a technology I want to briefly discuss how this can be used to look at cell-to-cell heterogeneity. Okay, so here is an example of using FISH to look at a single gene expressed for different cells. And you can see that this cell has a bright transcription site, many transcripts. This cell is dark, it's off, and then here these are some intermediate levels. So again, based on the same genotype, you can get different phenotypes. And well, like that, you might expect a lot of random variation based on this kind of noise. But oftentimes the results are the same, okay? Despite the fact that you have a lot of noise. So that's an interesting question. How do you actually obtain the robust result when you have such noise in gene expression? So there was actually a very interesting paper published 3 years ago. And so applying single molecule fission not only to culture cells or yeast, and they actually apply it to an animal. Okay? And it's about the decision making whether the animal can survive or not. And the title is variability in gene expression underlies incomplete penetrance. And I'll explain what that means. So this is the animal that they studied, C. elegans. Anyone has heard of this before? It is a very nice model system for studying development of biology and how fertilized egg becomes adult animal, because it's transparent and it's very small. It's very easy to grow. And you just feed the animal with E. coli, and then it'll be happy. So this is what it looks like. You have actually it has amazingly the number of cells is actually exactly known. Human body has about 10 to the 14 cells, but we don't know the exact number, right? But in the C. elegans, you know exactly how many cells are there in another animal. 959 cells. It's not an even number. Actually, this is for the female, and the male has about 100 more cells, but who wants to care about the male for this? Anyway, so you know exactly how things divide, and then some cells die in a way to actually result in the particular number of cells. And so in this topic, the interesting part of the animal is your intestine. The intestines are very important. Without this, you will die. Because people have mapped exactly how different organs develop each one of the cells. You also know how the intestine actually is developed. So there's a particular cell called yeast cell that divides multiple times to form the intestine. So you know almost everything about how the intestine is developed and what genes are required, to develop the intestine itself. So these are several genes that are required that are actually expressed at different time points. So as fertilized egg divides, initially this gene is expressed and then stops expressing here, and then later genes are expressed. Eventually, this gene called ERT2 is expressed somewhere down here, and then if that is made, then that becomes intestine. So if this gene has to be turned on, actually to start the process to make the cells into intestine-like cells. Eventually, that's why you have to turn on if you want to eat and digest food and so on. But to get that, these different genes are talking to each other. Arrow means that it will stimulate this gene, and in fact there's no inhibition here, but there's a very complex network that was mapped out by scientists. Usually, each line is a PhD project. So the idea is that if you have a mutation on this topmost gene, then you don't develop intestine, and then animal will die. So there's mutation, and the phenotype is the death. No intestine, and it will die. But that's not always the case. Okay? Some animals, even with a mutation, can develop the intestine. So that is called incomplete penetrance. Incompletely penetrant mutations reveal alternate cell effects. So many mutations are incompletely penetrant, meaning that not all mutant organisms display the mutant phenotype. So you make the same mutation there, then some animals will still develop intestine, meaning that these animals will express this ELT2 genes that I talked about. So there must be some other reason why some animals express and others don't. So I have a movie on C. elegans embryo developing. So how many cells do you see here? 4. You see nuclei here, 3, 4. So what they did was they collected the worm and then they fixed it so that you can add the probes. And then what they did, they squeezed the worm between the 2 cover slips, because if you make them flatter, then it's easier to image. Maybe it's better to like that. Okay, so it is 3D image, so you're looking at different planes. Now, blue is the nuclear staining, where the DNA molecules are, and then red ones are, red spots are single RNA molecules. Actually, this gene called ELT2. So when you have that means that you have the intestine cell that is properly activated. Just I missed that sound. Okay, so and then I think I have another one, so you can actually make a fancy movie. Right, you notice that you can see all of the cells, all of the nuclei, and you can count the number of nuclei and then use the stage of the animal development. But not all of the cells are expressed red spots, because only the intestine-like cells will do that. Okay. And also you notice that the animal is squeezed flat. Right, thank you. Good exercise. All right, so now I'm looking at wild-type embryos. Wild-type means that they are not mutants, normal animals. And then you see there are 3 embryos, and all of them have these red spots. ELT2 genes are being expressed normally. Now, if you have a mutation not on ELT2, too, but on this SKN1, where this is the most upstream gene, right, in this situ gene network. Then, interestingly, 3 of them have no ELT2 expressed, but this one has ELT2 being expressed at a high level. So this is what's meant by incomplete penetrance. You have a mutation, but the phenotype is seen only in 3 of the cells, not in all 4. So then, what they can do is to do multicolor imaging to look at the nucleus and also to look at many different genes at the same time. Then you can start to plot correlation between different properties. So in the case of the wild type, then what they're plotting here is the transcript count of this ELT2 gene. And then the x-axis is the number of nuclei. So as time goes on, and then embryos develop and grow and so on, the nuclei number will increase. So you use it as a pseudo time axis, and then this guy will eventually increase. But if you look at the mutant, it turns out that there are 2 populations. One of them has very low values of ELT2, and this one has actually levels similar to the wild type. So these are the ones that are actually showing ELT2 expression and testing development. It turns out that even these guys will eventually die. Okay, but so that is, and then because they can also measure the expression level of other genes in the network at the same time, they can actually deduce what is responsible for the distinction between the 2 groups. And there's actually a sharp threshold, and this also can do that kind of analysis. So this is really a nice example where noise in gene expression can make life or death decision for the animal. Not just a single bacterium, but for a real animal that has neurons and intestines and so on. So that was one example of gene expression tools that people are using widely at the single cell level. And another method can be called MS2 binding array. So if you want to now look at single mRNA molecules inside a living cell, you couldn't do that actually using the FISH technique because probes cannot go into the cell unless you kill the cell and make the membrane permeable. But so if you want to look at single mRNA molecules in living cells, then you can use a different method shown here. So the idea is that the blue is the RNA that you want to image, but then you do some genetic engineering to fuse that gene to this red sequence. The red sequence has this hairpin, and that hairpin is called MBS. It's actually MS2 binding sequence. So MS2 is a protein actually. That's that MS2 protein. And so if you put 24 copies of MS2 binding sequence fused to the genome of interest, then when this mRNA molecule, chimeric RNA molecule is produced, then that protein in the cell will bind to that sequence. But if that particular protein is also tagged with the fluorescence protein, then you can have up to 24 proteins, fluorescent proteins, decorating a single mRNA molecule. So again, you get a signal amplification to eliminate false positive and false negatives. And that allows you to actually track single molecules in live cells. Questions? So Ido Golding was a colleague here before he moved elsewhere. So I'm going to show you one movie that I got from him. So what they did was in the E. coli, they made this array of MS2 binding sequence, 96 copies. Okay? And then they also made MS2 binding protein fused to a green fluorescence protein expressed in the same cell. Then every time a new RNA molecule is produced, then that protein will bind, gang up on it, and that kinetic will appear as a green spot inside a living cell. So this is an example movie. You see E. coli cells, and whenever you see a green bright spot, that is due to a single mRNA molecule decorated with dozens of fluorescent proteins. And you can detect exactly at what point in time new transcript is produced and other information about this. Questions? There's a measuring tool that you can see a single mRNA molecule in a living cell. Now, let me compare the two technologies, single molecule FISH and MS2 binding array. Here, negative is that cells have to be fixed. You cannot measure things in live cells. So you can only take snapshots. You cannot take a movie. On the other hand, MS2 binding array actually is good because you can take movies, you can image in live cells. In fact, I'll show you a movie here where actin mRNA, you know what actin is? Actin forms an actin filament that forms the highway track for certain motor proteins like myosin 5, myosin 6, and so on. But in the brain of actually you need a lot of actins, but you have a nucleus of a neuron, but where the actin molecule is usually very far away. So actin mRNA has to be produced in the nucleus and then has to be transported down along the neurons, axons, and so on. So you can actually image that using this method too. Neuron axon, neuron's axon, and then you can see the transport of a single mRNA molecule inside the neuron using this method. Now, but there's downside for the MS binding array. Actually, you have to modify the gene. You cannot study the native cells or animal and so on because otherwise you cannot get the application. Whereas in a single molecule FISH, as long as you know the sequence of the gene, you can design the probes. So that is the term that people use, endogenous. Obviously, there's nothing that you have to do from outside to change the gene. You can just look at the way things are. So, in fact, because of this modification, you actually perturb the steady state level of gene expression. You may notice that when you look at this movie, you see single mRNA molecules in green dots, but normally mRNA molecules in the bacterial cells, they last only for a few minutes before they are degraded. So you see them, they are never degraded. Cells divide, each division takes, probably in this case like 40 minutes, but these green dots survive forever. They are immortal. Okay? The reason is that separate, one-handed mRNA with larger proteins, you are actually protecting it from degradation. So what it means is that you're going to, if you try to measure the steady state level by taking a snapshot and give you something about lactose and so on, your answer will be wrong because you're actually perturbing the system greatly in this manner.
Can you show the actin mRNA
Yeah.
on the neuron? Is that the mRNA that's coding for actin molecule?
Yes.
Can it be translated near its transcription site and then move along? So that's mRNA moving,
Yes.
not the protein.
mRNA.
Is the
Yes. So neuron. So because the neurons have very long processes, right? And mRNA is made here, but then the protein actin needs to be translated somewhere down here, so it has to be transported this way. Okay. And you are imaging it right there. Actually, this is a very amazing experiment that these guys did. What they did was to start with a mouse. Okay, you know what mouse is, and then they replaced the endogenous, actin gene by a gene, an actin gene fused to MS2 binding array. Many repeats. And then this data is taken from cells derived from that mouse. So that's called knock-in. Actually, most common one is knockout technology, but you can put the gene of interest into the thing. So but the mouse was born normally, and actually the mouse actually can reproduce. So I told you that this can seriously screw up the biological processes, right? But in the case of actin mRNA, somehow mouse was normal. So that's pretty amazing to me. There's nothing. So people can criticize this approach, where it's not really physiological. You can screw things, but when these people show a picture of a mouse, where the mouse is normal, mouse can make babies. So
So what is the driving force that moves the mRNA?
Oh, in this case, it's kinesin and dynein. Yeah, they move on the microtubules. So here, the transport is done by the kinesin and dynein monoproteins.
So the cargos are not restricted to proteins, I guess?
That's right. Cargos can be vesicles, membranes, compartments. Cargos can be mRNA molecules. Transport of mRNA in this direction is actually very known. Because you don't make mRNA down here, right? You make it here, then you have to send mRNA over here. In fact, I've just seen some data by somebody where this neuron will make a junction with another neuron, and that's how the neurons communicate to build memory and so on. If you stimulate the junction, then they can see that mRNA for actin somehow appears here. Okay. Stimulation recruits this mRNA because actin is needed right here to form a long-term memory.
When the mRNA is transcripted, the ribosome will bind to the mRNA.
Yes.
So that's affect these two methods.
Okay, so here it doesn't matter because you detect them only after you kill the cell, right? And then you don't make any changes to the genes and so on, right? But here you're making the changes to the gene, and the mRNA is lengthened and mRNA has additional proteins bound to it. But the fact that mouse survives, right? Mouse can be born and reproduce, suggests that translation is not affected too much.
Why can we not do fish in living cells?
Because the DNA molecules do not go in.
It's not.
DNA, so
not moving.
the probes cannot go in through the cell membrane. So what you do is that you start with the cell and then you kill the cell, and then you use chemicals to make holes on the cell membrane. So that
And then the hairpins in that method is built in.
built into the mRNA, yeah, built into the DNA. the gene itself.
So we can also do that in fish. We modify the DNA to make it produce
Fluorescent
proteins.
probes. Fluorescent probes. But
We cannot make DNA.
yeah, you can't make DNA with fluorescent. So if someone figures out how to decorate endogenous genes in live cells so that you can detect single transcripts, you get the best of the both worlds, then I'd be very happy.
Don't bacteria have a survival mechanism where if you put them in extreme conditions, they'll kind of uptake DNA that's in the surrounding medium? Can they not do that if there's a probe attached? Or is the interest here that you want to do it in multicellular organisms?
So, yes, you can convince the bacteria to take up some of the DNA molecules from the medium. Yes. So that's people have done something like this already in bacteria. You can actually put some even proteins with fat layers in the medium, and then they can be taken up by the cells, and you can use them inside the cell and so on. Another issue is that in the fish imaging, you actually have to rinse a lot because you don't want to have probes that are sticking and remaining inside the cell because they will contribute to the background. So you have to do extensive rinsing to remove proteins that are not bound to the mRNA. If the cell is not porous, then they will all stay inside. Right? That's another reason. Questions? All right, so I'm done with that part. We'll move on to genome engineering. Good. For those who came late, the final lecture will be given by Professor Schulten next Tuesday, 2 p.m. So that's a regular lecture, or not the Monday evening. Okay, the beginning of genome engineering. So that's the recombinant DNA technology. This is really a classical example because insulin is used to treat diabetes, and it used to be very difficult to obtain insulin. But now with the recombinant DNA technology, you can make it very easy. So what you do is that you have you have these scissors-like molecules called restriction enzymes that can cut DNA at a defined location. And then you can insert the DNA of your interest, in this case, the DNA coding for the insulin gene. And then you make this circular DNA molecule called plasmid and put it inside bacterium. And you can convince them to pick up the DNA. Right? Some cells, some strains are more willing to do so than others. Then this plasmid propagates, even the cell divides, and then actually you can convince the plasmid to express insulin. The bacteria will make insulin that is secreted and collect that insulin and then provide it to the patients. So I think insulin alone in the US, multi-billion-dollar business. So genome engineering actually can fulfill the need in several different areas. So I'm showing 3 different examples. Functional testing of genetic mutations, treatment of human diseases, engineering of synthetic biological systems. Yes?
Is that an actual lamp? It's so cool.
How much are you willing to pay? I'm ready for you. At least I didn't get the candidates wrong. Right? Okay, the first functional testing of genetic mutations. If you want to understand the function of a gene of your interest. What gene are you interested in most?
NS3 and NS5B with hepatitis C virus.
Okay. So then you can make genome engineering of the virus to remove NS3 protein from the viral genome and see what happens. Then if the virus does not replicate, then you know that NS3 protein is essential for the replication. So the ability to make changes in a defined way is really useful in understanding the functions of different genes. So that is like one reason. It's great for research. It can be great for treating human diseases. So one example would be, HIV virus target some receptors on immune cells, on cells in the brain. And if you know that a certain mutation of that receptor can fend off the HIV virus, then you can put that mutation into the stem cells of the human immune cells. Then if you do that genome engineering, then you can cure the patient. So that's one example. Engineering of synthetic biological systems. There are several good examples. You can use synthetic organisms like yeast that are engineered to produce precursors of malaria, anti-malarial drugs that can make the drug much cheaper. You can also engineer yeast to so that they can actually process farm waste, like corn stalks and so on, into ethanol for bioenergy production. So there are many actually reasons why genome engineering can be powerful and useful. And there's really actually a revolution going on in the last few years. That's why I want to talk about this. Okay, what is this?
White rice and golden rice.
Oh, you have golden rice?
I've heard of it.
You have anyone else has a golden rice? So this is the regular white rice, and this is GMO. What is GMO?
Genetically modified. Yeah.
Organism. GMO crop. Okay, raise your hand if you are for GMO. Raise your hand if you are against GMO. So if you're against GMO, then what do you eat? So you're against it. Okay. So in the U.S., you see, we are living in a very interesting place. Because if you look outside, we have cornfields and soybean field, right? I think over 95% of the crop is genetically modified, meaning that it actually, in the extreme sense, they have genes taken from other organisms to make them easier to grow. In fact, in Europe, these kind of crops are banned. You cannot sell them in Europe, but in the US, you cannot avoid it. Basically, 95% of what's produced in the field is modified. Not just that, if you buy meat, right? Then chicken, beef, and whatever, these animals actually they feed on corn and soybeans. Right? So this, and if you drink Coca-Cola, it comes from that genetically modified corn syrup. Right? That sugar inside. So you can be against it, but it's really impossible to avoid unless you go entirely organic. Organic crops, they are by definition not non-GMOs. So this is a GMO this season. So golden rice has a potential benefit. Let me explain here. So this is a paper published in Science in the year 2000 by these people. I think they have been working on this for 8 years until they work. So let's look at the title. Engineering the provitamin A, beta-carotene, biosynthetic pathway to carotenoid-free rice endosperm. Very technical expression. Let's look at the abstract. This must be rice. Uiza sativa, that's I guess Latin name for rice, is a major staple food. Is usually milled to remove the oil-rich layer that turns rancid upon storage, and especially in tropical areas. The remaining edible part of the rice grains, called the endosperm, lacks several essential nutrients, such as. So if you feed on this exclusively, then you get a deficiency in vitamin A, and causing public health problems in at least 26 countries, Asia, Africa, and so on. So what they did was they used a DNA recombinant technology to introduce enzymes that can be used to produce provitamin A, not on the oily part but inside, in the inner part. So that's an example. You know what this is?
Is that what Native Americans would have actually grown and eaten anciently?
Yes. So this is I think it's a teosinte, and so what you have now, modern corn, actually started from this. So that is actually the most ancient form, the more primitive side, and then so it looked like this, okay, and then Native Americans were desperate enough to groom this into something like that. So that is also genetic engineering, although they didn't actually do any, recombinant DNA technology kind of stuff, right? They simply, collected their, offsprings that have more desired features and so on. But it's amazing how different two can be. So when the Monsanto company they justify their modified crops, they say we are nature, or human beings have been doing this for many years already. So why is there anything special about what we do? This is a product that actually I buy from Monsanto for my garden. Roundup, have you used it before? Don't drink it. So Roundup's active component is this glyphosate. It looks like that. What does it do? Okay, phenylalanine, tyrosine, tryptophan, what are they? Amino acids. 3 out of 20 amino acids, because they are essential. Okay. And so in plants and some of the microbes, there's an enzyme called EPS-PS. Okay? That enzyme is required to make a precursor for these amino acids. That glyphosate, that Roundup's active compound that I showed you the structure of, comes and binds this enzyme and inhibits this. Okay? This means that the Roundup will kill every kind of plant. It will kill everything. So it's great for my garden where I don't want to have anything to grow. Right? So that I just spray. That I can even kill the not only weeds but my regular lawn grass invading that area. I can kill them no problem. That's why I use it for my garden. So Monsanto company discovered that in certain bacteria, that has an enzyme that actually is insensitive to that drug. So that drug, that molecule, cannot be used to kill that bacteria. So what they did, they took that gene and put that into the soybean. Okay, that means that you can spray Roundup in your field just once, and it kills everything else except for your soybean. In fact, I know how difficult it is to kill the weeds because there are many different kinds of weeds. Usually, you have to use special types of chemicals to kill different types, 3 or 4 different types each time, because pointed weeds, they are hard to kill selectively because you can also usually kill your lawn grass as well. But if you have Roundup, you kill everything else, then your soybeans will go just fine. So that, yes.
What's the decay, or not half-life, but how long does it take for Roundup to become inert when it's sprayed like this?
Probably not probably a few weeks. So, but you don't want to reseed the lawn after you kill everything, for another like a month or 2.
That just makes me wonder if the things, well, obviously if we can't drink it, it's poisonous to us too, and not just plants.
Well, no, I think it's not going to kill you. Well, I don't know. But I mean, it inhibits a plant enzyme, right? But probably it's not good for you.
Yeah.
It's not going to kill you in that way, but it's going to lead to cancer.
Oh, okay.
But it will kill the bacteria in our stomach, so that is not good.
I don't know. It's a special bacteria. Right?
Especially.
Yeah, so I guess it will kill other bacteria.
Yeah.
So bacteria that good for your yogurt. So that's what they do, right? Monsanto company sells their seeds, soybean seeds to the farmers. The farmers can then just do this once, and then so it's easier to grow, and the yield will be better because you don't have to fight against the weed all the time. So that's the reason why 95% of the U.S. farmers use this type of seeds. Of course, there's a lot of losses going on because Monsanto doesn't want the farmers to reuse the seeds. So I met a high-level Monsanto scientist once here on campus. He told me that, by the way, this is also true for the corn crop. So they continue to engineer their corn so that every year the yield increases by 2 or 3%. So for the farmer's point of view, it does not make sense to reuse the seed for 5 years because then your yield will be actually like 20% lower. Right? And that's a huge difference. It's really quite interesting. So how many bushels of corn that you can grow per acre? In the last 100 years, it increased by a factor of 15. Go out to the field when the corns grow, they are so close to each other, right? Spacing is, like this. They are taller than the meats. So they must have optimized the spacing to maximize the output. So you have top yield as a function of spacing, and that function was taken. You take a derivative and then set it to 0, and then you calculate the optimum spacing to this. Actually, Monsanto person told me that Monsanto has a database of entire Illinois. So every plot for cornfield, they know annual rain amount and sunlight and soil quality and so on. So if the farmers ask them to help them, then they'll use a robot. Based on the information, on the fly, they'll adjust the spacing. Okay. So it's pretty amazing. What you do in our labs is child's play. And there's a lot of money here because why do the farmers plant corn and soybean only? Because it makes money for them. It's a very productive crop. And they start using genome engineering technologies. Okay, so how do you actually do genome engineering? Usually, what you need initially is you need to introduce a break on the DNA at the desired location. So that's the first step that you must do. And that break, double-stranded DNA break, is induced by enzymes called the nucleases. The nucleases are enzymes that cut DNA or RNA. And then what happens is that you can cells will try to repair this break by connecting the two broken ends together. It's called non-homologous end joining between lots of different proteins. Or usually in the process, mutations introduced at the location. So it's, here they say indel mutation. What is indel mutation? Insertion deletion break. So sometimes when you connect it back together using this pathway, sometimes you can insert a couple of base pairs, or you can delete a couple of base pairs. The end result is that when you do that, then you can introduce a premature stop codon. So the stop codon is the three bases sequence that the ribosome recognizes, and it stops translating here. But here, usually in the regular sequence, there's no stop codon here. But if you insert or delete one or two, okay, then you shift that reading frame. Okay, because when you read the sequence, you read 3 at a time. But if you insert 1 or 2, then you can shift. Then you can produce a stop codon in a place where they use not your stop codon. Then when the cell is trying to make a protein, then it will just truncate right here. Then you can basically remove the gene or gene's product from the organism. So that's one way. But you can do something more sophisticated to replace certain segment of the DNA by the gene of your interest. So that is more powerful, but more difficult to do. But everything starts from here, basically making a break in the DNA exactly where you want. How do you do that when you have 3 billion base pairs in the human genome? That's a big challenge. Okay? So I'll tell you about 3 different technologies we have developed. The first one is based on one of the zinc finger DNA binding protein. And this is a really important paper. 2 structural biologists in Johns Hopkins University. They saw the structure of that protein bound to the DNA at high resolution. And let me show you a figure from the paper. You listen to many lectures by Professor Schulten, so you know how you can use VMD and computational tools to visualize protein structures and so on. But this was 1991. They didn't have any fancy program too. So this is a figure from the paper. Or this is what we use, the DNA and then proteins wrapping around the DNA and so on. But the key point here is that the protein has a module and then 2 amino acids recognizing 3 base pairs of DNA. And another set of amino acids recognizing another 3 base pairs and so on. So they could reduce the rules behind the recognition of the DNA base pairs in triple units. Then other scientists in 1996, they published this very important paper. Hybrid restriction enzymes, zinc finger fusions to fork on cleavage domain. So what they did was that they had the zinc finger binding protein that can bind to a certain DNA sequence, and then they fuse it to a nuclease. What it means is that then this protein will bind to a certain specific sequence inside the cell, and the nuclease will then cleave the DNA right there. So this technology is called ZFN, for zinc finger nuclease, because you have a zinc finger protein that binds to DNA fused to a nuclease. So again, the idea is that you have this zinc finger domain that records 3 base pairs of DNA. And if you have 4 of them recognizing different sequences, then this can be targeted to a specific DNA sequence, and then that's fused to a nuclease. Same on the right side. Then based on this, you can target 2 nucleases to precise location of the genome, and then make 2 cuts to make the representative break. And once you do that, then the rest can follow. You can introduce indel mutations, or you can also do genetic replacement and things like that. And people know a lot about the rules, how to make the mutations here to change the sequence, specificity and so on. So there's some way is to design this. Yes, and once you make the cut, you can do lots of genetic manipulations to the genome engineering. The New York Times, I think I read this one. New York Times covered this in 2009. The new way to edit DNA code for treating disease. So, HIV virus, and so there was a lot of excitement that zinc finger nuclease can transform gene therapy and so on. This is another one. So this is a company that has most of the patents on the technology, Sangamo. So a lot on genetic switch technology seeks to move into drug and so on. But now actually it looks like this technology is obsolete. 4 years ago this was like heralded as a big thing that would make lots of money for the company, help a lot of patients in the world, but now it became obsolete because now we have a new technology called TAIL. And then pretty soon this may become also obsolete because now we have a new killer on the block, CRISPR-Cas9 system. So I should tell you about these two now. So TAIL is a new DNA binding protein discovered very recently. So this is a plant tissue, and then certain bacteria grow, parasites grow on the plants. But what they want to do, they want to actually influence the gene expression pattern of the plant itself to become easier to survive for the bacteria. So what they do is this bacteria will insert a needle into the plant cell and then secrete small proteins into the plant cell. And these proteins are called TAL effectors. TAL stands for transcription activator-like effectors. So what it is that this bacterial protein, TAL, goes to the nucleus of the plant cell, binds the DNA to activate the transcription of certain genes that are useful for the survival of the bacteria. So TAL stands for transcription activator like effectors the bacteria produces to influence the host gene expression. But that protein has lots of amino acids, more than 1,000 amino acids, has multiple domains important for its function. For example, there's a domain called nuclear localization domain that makes sure that this protein will go into the nucleus, and then there's a DNA binding specificity domain that tells you to which DNA sequence the protein needs to go to, and then finally transcription activation domain. When the protein goes to bind to the DNA, then that domain will induce the transcription activation of that gene right there. But what's of interest here is this domain that confers the protein with DNA binding specificity, sequence specificity. It has many repeats, and each one domain, each one color, recognize one base pair of DNA. So there are two papers in 2009. Okay. So the people then discovered the rule coding governing the DNA binding specificity. What amino acid sequence of the protein is responsible for recognizing certain DNA sequence. There are two papers actually back to back. So I'm coming back to that protein itself. You have many repeats. But if you look at one single repeat here, then it has like 34 amino acids. And there are two of them, the first and second positions, that are hypervariable, meaning that everything else is essentially the same for all of the repeats. But these two amino acids are different between the repeats. So it's suggesting that these two amino acids are responsible for this DNA sequence recognition. So then they use the bioinformatics and other methods to decipher the rule. So if you have a certain amino acid combination, you will most likely recognize C. If you have this combination, most likely recognize A, and so on. So based on this information, then you can simply engineer this protein just by changing the DNA sequence, encoding for these amino acids. Then this can be used to target certain sequences specifically. So TALEN now stands for tail nuclease, and that was actually shown to be effective in genome editing and engineering. That's 2012. So this became actually a lot more popular because that used to cost more than $15,000 to engineer for one gene. This one costs $100. Finally, the CRISPR-Cas9 system, and here they found interesting protein and RNA in bacteria that makes it good for making yogurt. Okay. And then it turns out that you can hijack, you can reuse that protein and RNA system to make a cut on the human genome. And so this is the latest excitement. And so you can make that bacterial protein called Cas9, produce it in the human cell. You can also produce a certain RNA molecules that have the recognition sequence complementary to the human genome identification. That if you have that both, then Cas9 protein will bring that RNA here and then cut, producing double-stranded DNA gray. And then everything else is the same. So in this case, actually, it's even simpler to do so, and then TALEN. And now people are racing to make a library of the human genome, where each gene can be modified using this technology at a low cost and provide it to other users free of charge, things like that. So that is the latest in genome engineering technology, and it's clear that this guy is gone, and very likely this guy will be the winner over Talon. But in all of the cases, really what's important is the off-target effect. You want to minimize the off-target. You want to cut only the gene that you're interested in, not another location that is similar in sequence. So there's a lot of effort now going into engineering these systems to reduce the off-target effect. All right, so I normally stop here this time, but let me spend 5 more minutes if you excuse me, because I wanted to talk about the synthetic cell. So this is a picture of Craig Venter, Craig Venter, and he's the one who pioneered this the shotgun sequencing method. When human genome was being sequenced using the public money, he said, "Well, that's a waste of money. I have a better method. I can just raise much small amount of money and then do it on my own." Right? So it's a public effort sequence the genome of whom?
So then the person.
Yes, Jim Watson. And then Craig Venter Institute sequenced the genome of Craig Venter. Yes. But 3 years ago there was a science paper titled, I think this was like creation of a bacterial cell controlled by a chemically synthesized genome. Actually, I didn't see it first on the Science magazine. I first saw it on the New York Times. They have an editorial called "One Cell Forward," and researchers say they created a synthetic cell, quote unquote. So I was intrigued, and they even had they kindly put together some visuals to tell people what was actually done. So they found an organism, bacterium, whose genome is only 1 million base pairs long. E. coli has like 4.5 million base pairs, 1 million is much shorter. And then they were able to chemically synthesize 1 million base pair genome. And then they found another species of bacterium that is similar but not the same. And they started with the bacterial cell and then removed the DNA from the bacterium, and put the chemically made DNA to the empty shell. And that they showed that DNA can direct the growth and division of that shell to form colonies. And that's the idea of this kind of basically synthetic cell. So in principle, you can do this if you only know the sequence of the DNA, right? So you start from a database, and then you can make that into a living organism. So that was what they did. So the questions I had back then was that, like, okay, why is this a big deal? Okay. Is this going to be useful as a practical tool? Can I do anything differently in my own research? And how did they make a synthetic DNA of 1 million base pairs in size? Because if you try to synthesize the DNA chemically, you can do up to about 100 base pairs. That's about it. The yield drops so much, it's not practical to do so. And why did it cost them $40 million to do it? And it really costs the same amount to do it again. Actually, this paper reminds me of another paper published in Science maybe years earlier. And so 2002, if you recall, was the year when we had, was it 2001, when we had anthrax in the news. And someone sent spores of anthrax bacterium to the White House or some judges, newspaper offices, and so on. So the bioterror was in the minds of many people. So what they did was they chemically made poliovirus DNA, okay, without any virus, and then they could show that you can make live viruses just based on that chemically made DNA. Is that a big deal? Okay, what did they do was, they did some engineering to essentially eventually put together DNA that is 7,000 base pairs long, and then they put it into human cells, culture cells, and then they can make poliovirus. So that was the Science paper. But the Science published a commentary by my friend, whose eyes kind of was in black. And the title of the commentary was Not So Cheap Stunt. And he basically wrote that this is little more than a stunt, bankrolled by hundreds of thousands of dollars of tax dollars. And he says that, and Science magazine says that we are interested in tiny findings that are of importance scientifically. So he doesn't question the primacy of the finding, because that was when the anthrax was this and so on. But is it important scientifically? It turns out that the same group by Lemmer 20 years earlier, they showed that if you collect the DNA from the virus, nothing else, DNA alone from the virus source, and put it into human cell, then you can make a live virus. So he said, okay, DNA is DNA is DNA. So he wants to make DNA, then why is it surprising or profound or important to show that chemically made DNA can be the same as and so on. So the response from the magazine was that there leaves the claim that we have published a stunt, and we found about it that blood size figures would lead to a strong expectation of how the cell load at our measurement would come out. But proof of principle sometimes involves doing all the steps that demonstrate that the predicted outcome can actually be achieved. Certainly, this result is highly counterintuitive using a stunt. Do you think so? So anyway, so when I first saw this Craig Venter paper, I thought of this one, so I emailed Stephen Black, what do you think about the new one? I said, yeah, that's a stunt too. But it's very expensive and non-trivial stunt. So what they did, at least I think technologically it's really marvelous, it's amazing. So you have 1 million base pairs of DNA, right? They assembled that base DNA in 3 different steps. Okay? They first started with DNA molecules that are, so this is 1 million base pairs. So this is 100,000 base pairs long. And this is 10,000 base pairs long. So what they did was they first purchased DNA molecules from a company who produces DNA molecules that are 1,000 base pairs long already. That's, and then they did actually 3 steps to go to 10,000, 100,000, and 1 million. And I'm not going to talk about how they did these steps, but I want to just show you once how actually the company was able to make 1,000 base pairs of DNA when chemically you can only make up to 100 base pairs of DNA. this was farmed out to a company called Nanobio, and so that they'll probably charge about $1,000 per fragment. So if you ask a company to do 1,000 different, then that alone is $1 million. So the way you do it is that you have a solid phase surface, then you add a DNA molecule that is linked to the bead, and you rinse everything away. So each one is less than 100 base pairs of DNA. And then you add the next one, you link, and then rinse everything away. And you do it 10 times, then you get 1,000 base pair of DNA. That's basically what they did. And in fact, these days, most people do not actually try to collect the DNA from natural source. They just give a credit card and company to make it for you. That's actually much cheaper than do it in-house, especially in the industry. But in the university, graduate students cheap, so maybe it's still cheaper to do it in-house, but not much more so. So that's really taking over the entire equation. So I told you that sequencing technology is getting cheap, really is changing the equation a lot. Same is true in DNA synthesis. So you don't actually need to collect DNA from natural source anymore. If you have the sequence in the computer, then you can just make it. So that's where the real excitement lies. And what else can you do once you have these tools at your hand? Okay, thank you very much.